So microscopic examination of urine sediment. So urine volume for microscopy is recommended to be 12 milliliter, but the range of 10 to 15 or sometimes 6 milliliter for pediatrics is allowed in lab. When less than 3 milliliter of urine is available for testing, then there's no sedimentation, or there is no concentration of the sediment. So there must be just immediate microscopic examination, but there is no sediment. There is no concentration. So I don't think there is any echo. So I don't know because there is no echo with me or any source of that. Yeah, kindly you check your internet, you check everything. Okay, so centrifugation of urine. First of all, you have to well mix urine, and you centrifuge urine at 400 to 450 G, or relative centrifugal force, or between 1,200 or to 1,500 round per meter for 5 minutes, to avoid damaging the flagell-formed elements in urinary cast or cellular cast in urine. So sediment concentration and viewing. So after centrifugation, of course, the supernatant is removed using a pipette until 1 milliliter contained in the sediment. Then we have to suspend the sediment, and you have to avoid too vigorous agitation of urine in order to prevent from the damage of or breakdown of fragile or brittle formed elements, especially urine cast or wax cast can be broken down when into small pieces when too vigorous agitation is done. So then you pipette around a drop. It's just one drop, 15 microliter. It's just one drop of the suspended sediments onto a glass slides for analysis. That's for microscopy. So the urine sediment volume must fill the entire area beneath the cover slide without excess. So if distribution of formed elements is uneven after applying cover slides, a new suspension of the sediments must be prepared. Yes, so when you notice uneven distribution of formed element, for example, urine cells, so you must prepare another suspension. You have to apply another suspension, because that can, of course, always show the improper sampling. So reporting format depends on each lab, but of course, sometimes using a 10 low power field or 10 high power fields, you can enumerate urinary components. So some components can be reported in a qualitatively way, for example, plus system like mucus, +1, +2, crystals, bacteria, because you cannot quantitate them. But other components that you can enumerate, you can report them in a range between 0.2, for example, per high power field, 2 to 5 per high power field, for example, and 5 to 10 per high power field. So depending on what you see, but at least you have to go on 10 high power fields or 10 low power fields in order to report these components of urine. So now, apart from using light field microscope, also you can also enhance urine sediment visualization. You can enhance urine sediment visualization, of course, using other ways apart from bright field microscopy, because it can be difficult to see components, for example, mucus components, hyaline cast, bacteria, very small and low refractive index, as well as some other similar refractive index to that of urine cannot be seen clearly using bright field microscope. So that's why we need staining in order to increase visibility of those same refractive index to that of urine. So there can be staining, or you can even change and use phase contrast microscopy in order to enhance their visualization. So they also help to define look-alike entities such as monohydrate calcium oxalate crystals, which can resemble RBC and can be used to distinguish between mucus threads and hyaline cast. So staining techniques that are used, you can use Pravito stains. Of course, Pravito stain is one of the stain that we use for urine sediments or urine microscopy. So Pravito stains that we use, so they contain primarily chrysoviolates and safranin. So for example, the stanhemamabin stain. So stanhemamabin stain is a very commonly used Pravito stain, and this stain can enhance detailed view of internal structure of the WBC, for example, and also epithelial cells and RBC, mucus and cast, because in some cases they can look alike. So we need to distinguish them using a Pravito stain like stanhemamabin stain. So for example, that is a squamous epithelial cell stained with the SEDI stain, which is a modified stanhemamabin stain. You can see how then it can characterize internal structure and stain differently between cytoplasm and nucleus, and it can also show the presence of nucleus and even WBC around, and even RBC. As you can see, this is just RBC without internal granules, and this is a RBC, a rhizocytes or white blood cell with internal granules or components. So another good Pravito stain is also 0.5 solution of toluidine blue, can also be used as another Pravito stain. So the stain is a metachromatic dye that can stain cell component differently. So it has a power to stain differently a cell component. So that's why we call it a metachromatic stain. So then we can be able to distinguish between the nucleus and the cytoplasm of the cells, because they can become apparent with this toluidine blue stain. So it is also used to distinguish between cells according to their size. For example, leukocyte versus renal collecting ducts, because they can be similar size. So we can use a Pravito stain like 0.5 toluidine blue to distinguish based on their nuclear and cytoplasm outline. So they can be very helpful, because for example, leukocytes and renal tubular cells, they can look alike. They are the same size, and sometimes internal structure can look alike. So for example, as you can see this one, as you can see, you can be able to distinguish between these renal collecting duct cells. So with the internal structures as nucleated cells with the round nuclei, and also sometimes the cytoplasm is not so granulated as what you see in white blood cell. For example, these leukocytes, neutrophils with the toluidine blue stain, you can see that the internal lobulated nucleus can be seen using SEDI stain or toluidine blue stain. Toluidine blue stain. So because if you don't use the stain, it can be very difficult to distinguish WBC from renal tubular cells. So acetic acid can also be used as another solution. It's not a stain, but also adding acetic acid, 2 drops of acetic acid into a few drops of urine sediments. The nuclear pattern of WBC and the epithelial cell is highlighted, but RBC are raised. So this is very helpful to also identify WBC in urine, because normal WBC have a very low refractive index, almost a similar index to that of urine. So we can better use acetic acid in order to distinguish between the WBC, and even among them, and also have a chance to raise RBC, because they can also interfere with our total counts WBC. So other stains that we use apart from these stains that can be used to stain cells, other kind of stains that we can use are stains of fats or lipids. For example, Sudan 3 stain or oil red ore, they can also be used to confirm the presence of neutral fats or triglycerides. So triglycerides is also what we call neutral fat. In case we suspect neutral fats during microscopic examination, we can stain urine sediments to identify the neutral fats, of course, triglycerides. So these lipids can stain orange or red and may be found maybe as free-floating, as droplets or globules. Sometimes they can be found within renal cells or macrophage. So, and those cells, they are called oval fatty bodies. Oval fatty bodies. So when they are found in macrophage or other cells, and also these fats or triglycerides can also be found in cast, for example. They can also found in cast as droplets or oval fatty bodies in cast. So, so in contrast, otherwise, apart from these neutral fats, of course, triglycerides, cholesterol is not stained with fat stains. So cholesterol can only be confirmed using polarizing a microscope, but cholesterol cannot be stained with these stains or fat stains. So you can be able to show, of course, the oval fat bodies. As you can see, this is a cell. This is a macrophage. This macrophage that contains oval fat bodies. As you can see, the fat bodies can be seen, and you can report them as oval fat bodies of the triglycerides, of course, of the lipids, after staining with the Sudan 3 stain or oil red ore. So they can stain either as orange or red oval fat bodies in cells or free-floating cells. So apart from those other stains, we can also use a gram stain in urinalysis laboratory, but it is not really used. It is rarely used because when we want to do gram stain, we have to transfer our specimen to microbiology lab, because these are the two different labs. We have urinalysis labs. We have also microbiology labs can do different things. So we can transfer the specimen to microbiology lab for gram staining. Otherwise, gram stain is rarely used in urinalysis lab. So Prussian blue stain is also used as a route stain. It is used to visualize hemosiderin or free floating or in epithelial cell or in cast, so which we call urinary siderosis. This is, of course, iron granules in urine, and we in the form of hemosiderin because of oxidation of iron. So they will stain blue in urine. So they can be found maybe in some cells in urine, or they can even be found free floating. So after staining with the Prussian blue, we can be able to visualize them as blue granules on urine specimen. So we can also use Hansel stain. Hansel stain is a very, very good stain to identify eosinophil in the urine specimen. So Hansel stain contains methylene blue and eosin Y in methanol. So we use Hansel stain in order to identify eosinophiluria. Eosinophiluria, that is the presence of eosinophil in urine, especially in patients with acute interstitial nephritis. Of course, as you remember, acute interstitial nephritis is associated with hypersensitivity to penicillin, especially, and you find increased levels of eosinophil in urine sediments. Then it is mostly associated with acute interstitial nephritis, which is hypersensitivity to penicillin and derivatives of this drug. So when you identify these, so it is a good sign of renal disease, which must be quickly treated by cessation of drug administration. So we must cease, or we must stop drug administration in order to treat this hypersensitivity to this drug. So failure to treat, it can lead to serious permanent renal damage. So it can be unfortunate if you cannot be able to report the presence of eosinophil in urine, because it is very significant. So eosinophil in urine, sometimes you can't distinguish between other leukocytes from eosinophil. Sometimes you need to stain using Hansel in order to identify the eosinophil urea. So for example, this is how they look like. Of course, they would show the typical granules of eosinophils. Of course, they are orange, large orange granules in the cytoplasm of eosinophils. So that was about the stains, different stains that we can use in urinalysis laboratory to identify different components and different elements in urine sediments.